In this video, we will discuss about these roles of tumor-associated macrophages. In some of our previous videos, we have already covered how this tumor-associated macrophages uh, helps the proliferation and growth of the solid tumors. Specifically, we have discussed about pancreatic tumors. At the same time, it also helps in the immune surveillance. We will cover those things in slightly more in depth in this video. So let us quickly revise how this tumor-associated macrophages forms. So whenever the host body observed some harmful activity at any part of the body, it will recruit monocytes at the site of infection or at the site of these harmful activities. And these monocytes formations occurs at the bone marrow, from where these yolk sac progenitor cells will differentiate into the tissue-resident macrophages. And after that, it will, with the help of these blood vessels, it can transport at the site of infection. And they, at the site of infection, this monocyte can be differentiated into the two different, and generally differentiate towards this M1 phase, which has the anti-tumorigenic properties. And these are the cytokines, or these are cytokines that are responsible for differentiation of monocytes into the M1 phase. However, what happens with the solid tumors has this tumor microenvironment, and that differentiate the monocytes into this tumor-associated macrophages or M2 phase, and that phase M2 phase is responsible for the pro-tumorigenic properties. At the same time, it also helps in the early metastasis, and these are the cytokines that promotes this differentiation of monocytes to the M2 phase. So how this now we will cover in different aspects, or this different functioning of this tumor-associated macrophages towards this proliferation of the solid tumors. Uh, so what happens is that one of the things that it does is that it helps in the differentiation of this acinar to ductal metaplasia, or in other terms, it helps in the uh, spread of this pancreatic tumor cells across the pancreas. And how it will does is that it will promote the cells that has these KRAS mutations, and with the help of these cytokines, so that will leads to this pancreatic interepithelial neoplasia formation. So this is one of the intermediary stage of the PDAC, that is pancreatic ductal adenocarcinoma. And so uh, this is one of the way by which tumor-associated macrophages promotes the uncontrolled growth rate of the already mutated pancreatic tumor cells. And the next thing that it will does is that it will release some of the cytokines that will suppress the innate immune response. At the same time, it will also suppress the adaptive immunity. So it will not only work on the suppressing the innate immunity, but also on the adaptive immunity by inhibiting, for example, it will inhibit the innate immune response by providing a physical barrier and in order to suppress the adaptive immunity, it will inhibit the T cells activity, as these macrophages are overly expressed with PDL1 receptors. So it can interact with these PDL antigens on the T cells and uh, suppress the activity of the T cells, and immune thereby the T cells will not be able to act against the pancreatic tumor cells. So, so, and, so this is another way that the tumor-associated macrophages suppress both the innate immune response and the adaptive immune response. The next thing that it will work on is through this early metastasis. So in this case, what it will does is it will promote epithelial to mesenchymal transition by releasing cytokines that helps in this mesenchymal transitions. That some of the cytokines are mentioned here, and these cytokines will promote the proangiogenesis. Proangiogenesis means that they will have a additional supply. They will have the additional supply of the food that is necessary for maintaining the uncontrolled growth rate of the cells. And by releasing these cytokines such as this TGF beta is one of the cytokines that promotes the epithelial to mesenchymal transition. And combination of all these cytokines will lead to the early metastasis of the pancreatic tumors at the other site of the body, or early metastasis of the pancreatic tumors, or in other terms, it will the tumor will spread across different part of the body. The next thing that it will does is that it will suppress the chemo resistance or chemotherapy. It will leads to these tumor-associated macrophages cause chemotherapy resistance both indirectly and directly. In terms of indirect roles, what it will does is it will provide a physical barrier, such as it will remodel this desmoplastic stroma, so that this chemotherapeutic agent will not be able to interact with the pancreatic solid tumors directly. At the same time, it will suppress the immune responses also. Then immune response functions we already discussed in the panel B of the second figure. And in terms of direct role, what it will do is it will release some of the competitive agents, such as pyrimidine, to compete with this gemcitabine, which is the agent which is the agent by the in the chemotherapy that that is responsible to act against the tumor cells. So it will release pyrimidine that will try to negate or reduce the effect of this gemcitabine. At the same time, it will upregulate the cytidine, and that will decrease the sensitivity of this chemotherapy. And finally, what it will does is it will helps in the it will prevent the cell death that can be occur by chemotherapy by reprogramming the intracellular changes in the intracellular signaling pathway in a way that it will eliminate any apoptotic factors that are formed within the tumor cells. So these are some of the insights about how this tumor-associated macrophages works towards this proliferation and growth of the solid tumors, or and have some pro-tumorigenic properties. If you need to understand more about this tumor-associated macrophages in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.